Hi friends, welcome to Arnav's World. Today is the 6th episode in the biology series. So today we are going to discuss about the smallest organelle in a cell, the ribosomes. So ribosomes are macromolecular machines found within all cells that perform biological protein synthesis, which is also known as mRNA translation. So these ribosomes make proteins. So proteins are used by every cell. Every step of the cell, proteins will be used. So ribosomes are the most important organelle because of this protein synthesis. So a ribosome is made from rRNA. If you don't know, the R in rRNA means ribosomal, and ribosomes also have an adjective, that is ribosomal. Great. So these RNAs and proteins make up the ribosome. So therefore it is a ribonuclear protein complex. That's a very big name for just a ribosome. So only use a ribosome, not other things like that. So each ribosome is composed of one small and one large components called subunits, which were bound to each other. So prokaryotes and eukaryotes have different sizes of this. So in prokaryotes, the small one is 30S, and in large ones are 50S. And in eukaryotes, the small ones are 40S and the large ones are 60S. 10 difference. Yeah. Okay. The small one had mainly a decoding function and is also bound to the mRNA. M in mRNA means messenger RNA. That means the smaller one reads the mRNA to see what it has. Like it's reading this book, which is not actually there. And I am the mRNA, like that. I am the small ribosome, like that. Okay, the larger one has a mainly catalytic function and is also bound to the amino acylated tRNAs. The T in tRNAs means transfer. So I'll show you what tRNAs are. So here is a tRNA. And the aminoacylated ones have small amino acids on them. So if you don't know what these are called codons. So these codons have the genetic information or anything about the code. That's why it is called codons, with code in its name, like that. So for example, this has A, U, G. So this on tRNAs it is called anticodons, while on mRNAs it is normal codons. So for example, this is an mRNA down here. Yeah, this is an mRNA. So here for opposite of A is U, for opposite of U is A, the opposite of G is C. It will only connect if it is mixed with its anticodon. So yeah, and the information like here is related to the amino acid it is carrying. So this must be connected with only the amino acid. Yeah, a specific type of amino acid has a different type of code in the codon. Yeah. Okay, the ribosomal subunits of prokaryotes and eukaryotes are quite similar, with a big difference being only the size, which is 10S. Yeah. Proteins that are ribosomes can be found floating within the cytoplasm or attached to the ER. If you don't know, ER is the endoplasmic reticulum. I also made an episode about the endoplasmic reticulum. It explains about the rough and smooth. The rough one has ribosomes onto it like this, while smooth doesn't have it. That's why it is called rough. because then when we rub on it, it will be rough. But we can't touch it, so yeah. Okay, proteins that are formed from free ribosomes are released into the cytosol, which is the cytoplasm which surrounds the organelles like the nucleus, and is used within the cell, not outside or any other place. But the membrane-bound ribosomes, here membrane-bound means on the endoplasmic reticulum. The membrane-bound ribosomes produce proteins needed in some other organelles or outside the cell. That means it can be transported to other organelles or outside the cell. For example, these dots or the ribosomes. So it can be taken out or it can be taken here, and this is the endoplasmic reticulum. Yeah, that looks beautiful. Yeah. Okay, bacterial ribosomes will get synthesized in the cytoplasm, not in any other place. They randomly appear in the cytoplasm. Not randomly, but there is a small process in it. Yeah. But in eukaryotes, the process takes place both in the cytoplasm and the nucleolus. If you don't know, the nucleolus is a part of the nucleus where these things get produced, the ribosomes. Yeah. Okay, but mostly it happens only in the nucleolus, not in the cytoplasm, which is rarely. Yeah. Now I will explain protein synthesis. So ribosomes link amino acids together in the order specified by the codons of the mRNA molecules to form polypeptide chains. So these are the polypeptide chains. They are also called as peptide chains. Yeah. So this is the ribosome. This is the mRNA which was read by the ribosome, and this will now connect to the tRNAs. So here is an incoming tRNA which will get bound to the mRNA. So it already has an amino acid on top of it. So next, when it touches the mRNA mRNA's codon and the tRNA's anticodon will match together. Then the peptide chain will be connected by the ribosome. So then it would be ready. The protein will be taken. The body of the protein, which is made of amino acids, will be taken there like a long chain. But the tRNA will be just taken out. It will be thrown out like that. So it will be reused again to make more proteins, which is very useful for the body. Yeah. And the remaining thing, which is folding the protein, will take place in the endoplasmic reticulum. So this is for today about ribosomes. Bye.